ஹாய் காய்ஸ், வெல்கம் டு லேர்ன் வித் கோக்ஸ். இன்றைக்கி வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி ஒரு ஆட் பார்த்துடலாம். BuyMote ஷாப்பிங் ஆப். இது ஒரு ஆன்லைன் ஷாப்பிங் ஆப். இதில் க்ராசரிஸ் எல்லாம் அவைலபிளாக இருக்குது. மற்ற ஆப்ஸோடு கம்பேர் பண்ணும்போது இதில் குவாலிட்டியும் அதிகமாக இருக்கும், அதே மாதிரி டிஸ்கவுண்ட்டும் கிடைக்கும். ஸோ ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோர் ரெண்டுலேயுமே இது அவைலபிளாக இருக்குது. டிஸ்கிரிப்ஷனில் லிங்க்கும் டீட்டெயில்ஸும் இருக்குது. செக் பண்ணிவிட்டு இன்ஸ்டால் பண்ணி பை பண்ணிக்கோங்க. இது ஒரு ரொம்ப யூஸ்ஃபுல்லான ஒரு ஆப். அண்ட் இதில் ஒரு இம்பார்ட்டண்ட் விஷயம் என்னென்னா டோர் ஸ்டெப் டெலிவரியும் அவைலபிளாக இருக்குது. ஸோ மிஸ் பண்ணிடாதீங்க, யூஸ் பண்ணிக்கோங்க. So today we are going to see about biochemical test, mainly the principles and procedures like Indole, Methyl, Voges-proskauer, Citrate, TSA, Urease, oxidase and catalase. Initially we have to see Indole first. So today I am explaining with the images only. Okay, now we are going to see the procedure first. Procedure is nothing. We have to prepare tryptone broth medium in 2 test tubes, one for control and one for our purpose, and incubate our sample organism into that broth culture. After 24 hours we have to add the indicator, that is Covax reagent. If cherry red color ring or pink color ring is formed on top of the broth medium, it indicates the positive result. If there is no color ring formation, it indicates the negative result. Now the principle behind this is we have inoculate the organism into the tryptophan broth, okay, which means the broth contains tryptophan amino acid. Some certain microorganisms are able to produce tryptophanase enzyme that break down the tryptone amino acid into ammonia, pyruvate and indole. Okay, these 3 compounds present in that broth tube. While adding the indicator, which means Covax reagent, that Covax reagent contains para-dimethyl aminobenzaldehyde. Okay, these organism is produced the indole, means this indole is react with that para-dimethyl aminobenzaldehyde and form a cherry red or pink color ring. Okay, this is the principle of indole. Now the next one is methyl red test. Here the procedure is we are using the MRVP broth medium to inoculate the organisms, our sample organisms. After incubation 24 hours, all biochemical reaction are done by the bacteria. Now we have to add the indicator, which means methyl red here, that is the indicator. Okay, we have to add 5 drops of methyl red and wait for 30 seconds. If red color formation indicates the positive result, if pale yellow color indicates the negative result. Then the principle is to detect the strong acid production. Here the MRVP broth medium contains glucose phosphate broth, that is glucose phosphate broth. Here the organisms ferment the glucose and produce some strong acids like lactic acid and formic acid. Here mixed acid fermentation will done, and by this fermentation the pH is lower to 4.4 or below that. Here now we have to add the indicator. Methyl red is the indicator here that shows the glucose fermentation or acid production is done here or it. If acid production, mixed acid production is done, red color positive result will be shown, and no formic acid or lactic acid production is there, no positive result, no color change will occurs. And now is the Voges-proskauer test, which is similar to MR test, which means here the broth used also contains glucose phosphate broth, which means MRVP broth medium. Here we have to detect the production of neutral end products. In MR we have to detect the strong acid production, but here we have to detect the neutral end products produced by the microorganism, which means for example acetone. Acetone is a neutral end product here. Okay, if the organisms ferment the glucose phosphate broth, it will produce the end product like acetone. If the organism growth in glucose phosphate broth, that must uses this pathway, which means butylene glycol pathway is used by that microorganisms and produce the end product like acetone and 2,3-butanediol. These two are the end product produced here, and after we added the indicator, that means alpha naphthol and 40% KOH. This end product acetone is present in the medium, means this acetone will oxidize into diacetyl. While adding the indicator, this acetone is oxidized into diacetyl, which reacts with the guanidine compound. This guanidine compound is present in the medium, which shows the color change here. If positive means it shows brown, pink or red color in the medium. If no positive result, negative means no color change occurs. But the procedure, now we have to see the procedure. What is the procedure? Is same to MR, which means MRVP broth medium is ready, sterilization, and after sterilization we have to inoculate the given sample microorganisms and incubate for 37 degrees Celsius for 24 hours. After that we have to add the alpha naphthol and KOH indicators, and we have to wait for 10 minutes to observe the result. These are the principle and procedure for VP test. And next one is a citrate test. Here previous MRVP, Indole or broth cultures, here we have to prepare the agar slants to see the result. So here we have to take the cement citrate agar and sterilize it and make a slant. After that inoculate. We have to inoculate here using the inoculation needle. We have to dip inside the bud and stick across the slant. Our culture is inoculated now, and we have to incubate at same 37 degrees Celsius for 24 hours. After 24 hours, blue color, Persian blue color appears, indicates the positive result, and no color change indicates the negative result. If the blue color formation detects the organism, our given organism takes the citrate as a sole source to grow. Okay, here what is done in the principle means microorganism use citrate as its sole source of carbon and ammonium salt as its sole source of nitrogen for growth. Okay, some microorganisms produce citrate permease enzyme. It utilizes citrate to grow in that medium. Okay, here in cement citrate agar, 3 components are here to grow that citrate permease enzyme producing organisms. What are they 3 means? Sodium citrate, it acts as a carbon source. Ammonium dihydrogen phosphate acts as the nitrogen source, and bromothiamyl blue acts as a pH indicator. Okay, these 3 will act as a sole source for that microorganism to grow in that medium. Okay, this is the procedure and the principle of cement citrate agar, which means citrate test. And next is the catalase test. Here the procedure is we have to take the culture in the slide. Okay, in the slide, and add hydrogen peroxide H2O2 in that slide. If effervescence, that means bubbles, is formed, indicates the positive result. If there is no bubble formation, indicates the negative result. This slide test may also done in tube. Okay, now the principle of this catalase test is some microorganisms have the capability to produce catalase enzymes. If that culture contains catalase enzyme producing organisms, the catalase enzyme will be present in that microorganisms containing culture. That catalase enzymes go and bind with the hydrogen peroxide and break down the hydrogen peroxide H2O2 into H2O, water, and O2, oxygen. This H2O2 will break down into H2O and O2. Okay, in this reaction indicates the bubble formation. Okay, This is the principle and procedure of this catalase test. And next is the oxidase test. This is very simple test because oxidase disc are available. Take that disc in a petri plate or a slide and take our sample and place one drop inside the center of the disc. If purple color formation indicates the positive result, no color formation indicates the negative result. This is the procedure. Then the principle is some microorganisms produce cytochrome C oxidase enzyme. Cytochrome C oxidase enzyme. This enzyme involves in the electron transport chain cycle within that organism. Okay, electron transport chain cycle done in that organism for that to react with that cytochrome C oxidase enzyme will produced by that organism. Now in that oxidase disc, paraphenylene diamine dihydrochloride is incorporated as a artificial electron donor. Now the organism contains cytochrome C oxidase enzyme that will react with this disc containing paraphenylene diamine dihydrochloride and produce dark purple color. Okay, this is the principle. And now Urease test. Here the procedure is we have to prepare the slant by the agar Christensen Urease agar or urea agar. We have to prepare slant after sterilization and inoculate and incubate for 37 degrees Celsius at 24 hours. After that, pink color indicates positive result and negative result indicates yellow color. Okay, here the principle is some microorganism produce urease. Okay, that urease will hydrolyzes the urease enzyme. That urease enzyme hydrolyzes the urea into ammonia and CO2. Okay, that leads to an alkaline shift in that pH. The pH should modify into alkaline condition that will changes the color from yellow to pink. Okay, this is the principle. And last is a TSI, triple sugar ion test. The procedure is very simple. We have to take the TSI agar and sterilize it, inoculate it and incubate at same 37 degrees Celsius for 24 hours. After that, various types of result are observed. Here we are incorporated 3 sugars in same medium. So different results are available here. First, the principle is to here mainly to differentiate the gram negative enteric bacteria. We have to use the TSI test. Okay, here fermentation of carbohydrate is done. Carbohydrate means nothing, 3 sugars, glucose, lactose and sucrose. Okay, by this fermentation gas production will occur in some cases. Okay, and H2S production also identified here. Okay, here some in TSI agar medium some nutrition sources are incorporated. They are 3 sugars, that is glucose, lactose and sucrose, and peptone as protein source, and phenol red is added as for pH indicated, and sodium thiosulfate and ferrous sulfate is added to detect the H2S. This is for H2S detection. Okay, now see in this image, control becomes plain red or dark pink in color, and here acidic condition indicates yellow color and alkaline condition indicates pink color. Okay, and H2S production indicates black color, and gas production indicates crack in the agar slants or break in the agar slants, or upliftment of the agar slant is considered as gas production. Okay, see the second test tube here, red slant, red butt, and no gas formation and no H2S production, which means the slant and butt both are alkaline condition, which means fermentation of 3 sugars is occurs occurred here, and no gas, no crack, which indicates no gas formation, and no black color indicates no H2S. In third slide, red slant, which means alkaline slant, and yellow bud detects acidic bud, no gas and no H2S. In the fourth test tube, we are clearly see both are acidic slant and acidic bud, and crack in the center of the slant and bud, which indicates the gas production. So gas is produced here by fermenting the sugars, and no H2S production because no black color. In fifth test tube, H2S production is occurs, and some cracks in the agar, which means some gas is produced here, and acidic slant and acidic bud, no alkaline here. And in last test tube, slant is alkaline and acidic bud, and crack here mentioned that gas is produced, and black color indicates that H2 is produced. Okay, these are the principle and procedures. Now the biochemical tests are over. Thank you for seeing these videos. If you understand these videos, please like my video and subscribe my channel. Thank you.